Well, it's the window's 8 to 10, but it's only an hour exam. So we decided just to let you guys sleep an extra hour. Or study an extra hour. Okay. All right. We good here? We good on the Zoom? I think so. All right, so we've got oops, where did my thing go? Right. Okay, so final exam is coming up. Okay, so we've got 2 lectures left today, which is going to be neurotransmission and neuroplasticity. Okay, and then Thursday we'll do how the motor system works and a very quick rundown of different sort of parts of the brain. Probably more on motors than on parts of the brain. But to be clear, okay, for the final exam that's going to be Wednesday, okay, the 7th at 9 to 10 a.m. You can show up at 8. It's on the schedule as 8. You can show up at 8, just relax, get settled in, but the exam won't start till 9 because it's only an hour long. So remember that exam is not cumulative. It's only going to be on the content covered in the last 8 lectures. Okay, so what is that? It's 1 month of lectures. It's endocrine system. It's sexual reproduction. It's the brain and the motor system. Okay, and we'll tell you where to go in an announcement coming up shortly. I'm going to host a practice exam this afternoon. I'm going to write my exam the next day or 2. And then I'm going to make a study guide for that exam in the next day or 2. So I will have that posted by the end of the week, hopefully before that. I will do a exam review session. What day is best for an exam review session? Would you prefer that to be on the weekend, on Monday of next week? Who would like it to be on a Saturday? Who would like it to be on a Sunday? Okay, we got, raise them high, raise them high. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10-ish. Who would like it to be on Monday? Okay, Monday's the winner. Okay, so we'll do a Monday. And that'll just be a sort of a, long form Q&A. And, whatever we want to talk about is good. Any questions on this stuff? Okay, let's do it. I should turn this on, huh? Okay, there we go. Anything in the chat? Oh, we gotta admit people, admit all. I wish you could auto admit. Okay, here we go. So, kind of boomy. Bring that down. So, last time we started talking about this, and oh my God. Okay, we started talking about neurotransmission last time, and we didn't get very far. So let's just go over it again. Okay, so what are you looking at here? Right? Electron micrograph. There's a synapse. Where's the synapse? The synapse, I mean, there's a lot of things going on in this electron micrograph. We've got slices of axons here. How do we know they're in an axon? Because they're wrapped around a bajillion times from oligodendrocytes. Okay, or Schwann cells, depending on where you are. This is a central synapse, though. Then we've got, probably these are little pieces of dendrites from other neurons here. And then we've got 2 different sides of a cell. There's a connection, not a direct connection, not an actual fusing of the plasma membrane, but there are scaffolding proteins here that are holding on to the presynaptic side and the postsynaptic side are holding on to each other, keeping them closely aligned so that the active zone, the place where the neurotransmitter is going to get released, okay, which are in these little vesicles here, is going to be directly deposited into the synaptic cleft. This synaptic cleft is really narrow, okay? It's on the order of 25, 30 nanometers across. Do you think the fusion is going to do a good job at going that distance or not? It's going to be really good. Okay. Diffusion works faster, the better over very short distances, right? If you're going nanometers, diffusion is incredibly fast, right? You're going to get across there in a microsecond. Okay, very fast. Okay, so we're going to dump this little bag of neurotransmitters out there. How many neurotransmitters are in the bag in the synaptic vesicle? About 5,000 molecules. They're going to come out, they're going to land on this postsynaptic terminal. Okay, and this is where we have this density called the postsynaptic density. Okay, and along there you have studded along there, you're going to have receptors, ligand-gated receptors that are going to bind to the neurotransmitter and open up. Okay, there could be different types of neurotransmitter receptors there, though. Okay, you could have glutamate receptors, you could have GABA receptors, you could have acetylcholine receptors, you could have dopamine receptors, you could have norepinephrine receptors, you could have neuropeptide receptors, you could have any sorts of receptor there. Okay, but the important thing is there's a directionality of flow of information here, and an ability for one cell to pass information and influence the membrane potential of the postsynaptic cell via a chemical transmission, okay? And that chemical transmission is actually very fast. The entire process from release to actually starting to get your depolarization only takes less than a millisecond, about 1 millisecond, okay? So when you think of chemical transmission as like a slower process than an electrical transmission down an axon, it's true in that like you can't send a chemical signal a long way, right? You can't send this chemical signal a long way in a short amount of time. You have to use your electrical action potentials to do that. But if you're just going a short distance to go between one neuron and the next neuron, since it's a tiny little distance, diffusion works great and you can transfer the information very quickly. And there's a number of advantages to doing it like this, rather than just letting the electrical signal pass from one to the next. Okay, and we'll talk about those advantages in just a second. So what are these synapses, right? These synapses are the communicating connections between two neurons. Okay, here we have now immunostained a hippocampal neuron in a dish, probably. Maybe it's in a slice, I don't know, of brain. And we've here stained synapses. In green are the presynaptic boutons, and in red are postsynaptic proteins. So we've used two different colors. We've used immunoglobulins that are conjugated to a dye. The immunoglobulin, it binds either to a presynaptic protein or a postsynaptic protein in green or red, okay? And you can see there's a lot of yellow here. What's that yellow mean? It means that the red is overlapped with the green. The presynaptic proteins are always found in conjunction with the postsynaptic protein. Those two things are aligned so that the neurotransmitter is getting released in the right place to actually have an impact on the receiving cell. Okay. So the word synapse itself, which means connection, was coined by this physiologist, Charles Sherrington, back at the end of the 19th century. And here he is talking to John Eccles, who figured out that the actual mode of transmission was chemical. Eccles actually thought that it was electrical. He spent his first decade of his major research career trying to prove that it was an electrical connection between the cells, and eventually he gave up, changed his mind, decided it was chemical, and then proved it was a chemical synapses, okay? So we can broadly classify synapses. Now, the interesting thing is Eccles wasn't completely wrong, okay? There are two classes of synapses, okay? There's electrical synapses and chemical synapses. There are a few places in the brain and in the peripheral nervous system where electrical synapses dominate, okay? Those are where you're going to have direct electrical connections from one cell to the next. How are we going to do that? Gap junctions, we've seen them before, right? These are just pores that are going to be aligned between two cells and allow ions to flow past, okay? But the most numerous, by far the dominant form of synapse in the central nervous system, the brain, is going to be, and on the muscles, okay, from the nerves to the muscles, is going to be chemical transmission, okay? Chemical synapses. So let's briefly spend one slide on electrical synapses. How might they work? In the electrical synapses, we have a direct connection between our cells. It's going to be by gap junctions, and these are non-selective pores that will allow any sort of ion to pass through. Okay. And also, those are going to allow passage of small molecules to go through. So this allows pretty rapid signaling. You can get the ion can just come in the presynaptic side, flow to the postsynaptic side, or flow from one cell to another cell. This is sort of what you might see in cardiac myocytes that are going to allow electrical waves to propagate throughout their tissues. Okay. Now, one of the things about this, though, is that if this is almost like a resistance. There's paths for ions to travel, right? So electrical activity can go from here to the next, but there's also a resistance to that flow, right? There's not an infinite number of pores. So the sort of amount of electrical signal that you get on the postsynaptic side is always going to be attenuated. It's going to be a little bit less than on the presynaptic side. Okay. And so if you want to regenerate a signal, then you're going to want to use something other than an electrical synapse. Now, how do we know if a synapse is an electrical synapse or not? We can inject a dye from a pipette. We can poke one cell with a pipette full of a dye. This one's called Lucifer yellow in this example. Okay, and here we're in the retina because the retina does lots of signaling like this. Okay, and we fill up this cell with the dye and just wait. We wait an hour. And what do we see? We see other cell bodies and first we see all the processes of that cell. And then we see all these other cell bodies that are starting to light up. This is how we know that it's an electrical connection. One of the ways we can know it's an electrical connection, because there is a direct passageway between one cell and the neighboring cells. That is not the case in chemical synapses. There is no direct connection. The chemical synapse has to release a molecule, bind to the postsynaptic cell, and then transduce a signal on the postsynaptic cell. So if the retina area, this cell had no electrical connection with other cells, it was all chemical, we would only see an outline of just that one individual cell. Okay. Let's look at our chemical synapses now. This is the sort of structure that we just saw. Remember, that electron micrograph that we took a slice through, we basically took a slice through it just like this. Okay, we see our synaptic vesicles here. We see our mitochondria, our powerhouses of the cell. They're making lots of ATP. This process that releasing vesicles, fusing it with the membrane is highly energy intensive, right? And of course, after you fuse with the membrane, there's not an unlimited number of synaptic vesicles that you have. You can't just keep dumping it out in the membrane. The membrane will get bigger and bigger and bigger. We've got to take those synaptic vesicles back and we have to recycle them. Okay, so they're going to get recycled sort of up here. Okay? And so that whole process requires energy. That's why we have the mitochondria sitting there at the synapse. Okay, so the chemical synapses have no intracellular continuity. We've got this small space between the presynaptic neuron, that's the side that's going to release the neurotransmitter, and the postsynaptic neuron, that's the side that's going to receive the neurotransmitter. Okay, and in between we have that synaptic cleft. So current is going to flow across this postsynaptic membrane here. Okay, we're zooming in here. It's going to flow only in response to the binding of a neurotransmitter to some chemical that's being released by the presynaptic cell. So we're going to release this neurotransmitter. There's our 5,000 molecules of this neurotransmitter coming out. There's dozens or maybe 100 or so receptors on the postsynaptic side, maybe more. Okay. These are going to bind to those receptors. Okay. If you have all of the binding sites on that receptor full, they're going to open up. These are ligand-gated channels. They're going to open up, and then cations or possibly anions, depending on the channel, will flow through. Okay. And of course, when you open up a channel, you're now going to have a strong influence on the membrane potential, right? Because the membrane potential, as we learned from last week, the membrane potential locally is always going to be dependent on two things: the concentration of different ions inside and outside of the cell, right, which is relatively fixed, and two, what is the permeability of the membrane to those different ions? because they'll start moving across the membrane until they move towards their equilibrium potential for potassium or chloride or sodium. So by opening channels here on the postsynaptic membrane through the release of these neurotransmitters and binding of them, we cause a change in the local membrane potential. Okay, okay, so let's look at some features. Why would we use a chemical synapse rather than an electrical synapse? One reason is signal amplification. Okay, we have a single action potential that comes down the axon. Okay, it's going to hit a presynaptic terminal. It's going to hit more than one presynaptic terminal. In fact, it's going to hit every single presynaptic terminal on that entire axon that's going possibly all over the brain. Okay, so it's going to come in there, zip, get this little pulse of electrical energy, and you're going to release a single vesicle of neurotransmitter, maybe more than one. Sometimes you might fail to release a neurotransmitter or a vesicle, but on average, you're going to release about one vesicle at any one of these synapses, okay? That's going to dump thousands of neurotransmitters onto the postsynaptic cell, and that can open hundreds or thousands of channels in the postsynaptic cell. So that single vesicle can potentially depolarize a postsynaptic cell and cause it to fire an action potential. It's going to depend on how many receptors are there. Usually a single vesicle, at most places, a single vesicle won't be enough to fully depolarize a cell to cause an action potential. But together, they're going to work together to be able to drive another action potential, a chain reaction that's going to move down whatever neural circuit is connected there. And really, your thoughts are just the chain reactions of these electrical signals propagating across chemical synapses. So we've got a signal amplification. So a single action potential, a single vesicle can make a big response on a postsynaptic cell potentially. Two, it's rapid and transient signaling. So the receptors on the postsynaptic side, they have relatively low affinity for neurotransmitter. Okay, they require a high concentration of neurotransmitter to be released onto them for them to open up. So they only open up for a few milliseconds, maybe 10 to 50 milliseconds long, okay? It's a pretty short amount of time. The transmitter is going to be diffusing away, okay? It'll unbind to the receptor will close, and your membrane potential will fall back down to our normal resting membrane potential, okay? So that allows, though, because the signaling lasts like 10 or 20 milliseconds, okay? That allows you to send messages over and over and over again, right? You can send it after 20 milliseconds, you're ready to send another message potentially. Okay? So it's a pretty rapid pace at which, a rapid cadence at which you can transmit information from one cell to the next cell. And finally, neurotransmission dramatically increases the versatility of the communication between two neurons. If you have an electrical synapse, all you're going to do is send a kind of attenuated copy of whatever's in the presynaptic terminal to the postsynaptic cell because it's going through the gap junctions. Boring, okay? In a chemical transmission, you have a great selection of different neurotransmitters you could release and a great selection of receptors that can respond to that neurotransmitter. And so you can get a greater diversity of postsynaptic responses. And that can be tailored not only within a cell will have multiple different classes of receptors on it. So you might have receptors that respond to glutamate that cause depolarization and excite the cell, receptors that respond to GABA, which open chloride channels, which will inhibit the cell, receptors that respond to some like dopamine or acetylcholine that maybe manipulate the overall excitability of the cell, okay? Now, so within a single neuron, you can have many different receptor types, but also in different tissues, you can have different receptor types, even for the same neurotransmitter that cause distinct outcomes on that postsynaptic cell. We saw this with epinephrine, right? Epinephrine was coupled to two different G protein coupled receptors, one that excites, or one that causes vasoconstriction, one that causes vasodilation, depending on whether or not you're in skeletal muscle or in the gut. Another example here would be acetylcholine, ACH. On the heart muscle, Acetylcholine released onto the heart muscle is going to slow down the heartbeat. If you release acetylcholine at the neuromuscular junction, where a nerve is innervating and making synapses onto muscles themselves, that causes a strong depolarization and the ultimate contraction of the muscle. So same chemical, different outcomes depending on the receptor type. Any questions about chemical or electrical synapses yet? Okay, so let's now look to see what are the key steps in this process of neurotransmission. Okay. What happens? Okay. Well, first of all, we've got to have an action potential, right? Like we talked about sort of the action potential last time, right? We've got this wave of electrical activity. It's going down the axon, maybe via saltatory conduction if you're myelinated, maybe not if you're not myelinated. It hits the presynaptic terminal, but we stop there. We don't know what happens, okay? How do we actually send that signal to the next cell? 6 steps. Step 1. The action potential comes into the presynaptic terminal. Okay. So what is that action potential doing? It is really a wave of electrical membrane potential that is depolarizing it. We're going from negative 70 millivolts. We're going up to maybe plus 30 millivolts. Come back down. What will happen during that depolarization? Well, it's going to depend on what sort of channels are along the axon and in the presynaptic terminal. Now, we talked about these voltage-gated channels that allow an action potential to take place. What are the 2 voltage-gated channels that are important for that? Sodium, what does sodium do? Depolarization and? What's the potassium do? Repolarization, right? Okay, so those voltage-gated channels allow the action potential to take place, but you can also have other voltage-gated channels. Okay. One of those voltage-gated channels are voltage-gated calcium channels. Okay. So, and calcium is an incredibly important signaling molecule, the single most important signaling molecule in all of your cells. And why is that? Okay, it's the only ion that really, the reason is it is the only ion that ever appreciably changes its concentration inside or outside of the cell. Really, actually inside the cell. Okay. All those other ions, sodium, potassium, okay, they're not like, they're at pretty high concentrations. Moving them across the cell a little bit to change the membrane voltage doesn't really change their overall concentration that much. Calcium, on the other hand, calcium, the inside of your cells, cytosols of your cells, have got calcium pumps working overtime all the time, trying to keep the calcium outside of the cell. We want very low levels of calcium inside the cell. Instead of millimolar levels like you have with everything else, calcium inside the cell is on the nanomolar level. Okay. So if you open a voltage-gated calcium channel or any calcium channel, we've got millimolar levels of calcium outside, we have nanomolar levels of calcium on the inside. What do you think is going to happen? You open up a voltage-gated calcium channel, calcium is going to come in, and that is actually going to cause a substantial increase in the concentration of calcium inside the cell. That is the only ion that is ever going to change their concentration appreciably to an action potential. Remember that. Okay. And that is why it's such a good signaling molecule. So the wave of electrical activity from that action potential comes down to the presynaptic terminal. It opens up the calcium channels, these voltage-gated calcium channels. It doesn't open them for long. They're only open for as long as the depolarization lasts. But that short amount of time is sufficient to cause an injection of calcium into the presynaptic terminal. Okay. Now notice that action potential is not going here. It's not opening up voltage-gated calcium channels in the presynaptic cell. Right? All it's doing, it can't get there. It can electrical wave has to only stay within the plasma membrane. Okay, the plasma membranes don't touch here. Okay? So the wave of electrical activity comes in. We open up the voltage-gated calcium channels. Calcium rushes in, and what's it going to do? It is going to bind to a bunch of different things. Okay, but there is a whole sort of apparatus of protein machinery that it binds onto in the presynaptic terminal that will move synaptic vesicles over to the docking zone here and cause diffusion of that vesicle to the presynaptic plasma membrane. Upon fusion, what will happen? All the stuff that was in the vesicle that was now contained within this like little, bag is now fused with the outside surface of the cell. So that gets released. It gets dumped essentially into this synaptic cleft. Now, very rapidly, because we're only talking about nanometer distances, that is going to diffuse over here to the postsynaptic side, bind on to ligand-gated ion channels. There could be some other things that it can bind onto too, right? You could bind onto metabotropic channels or metabotropic receptors, G protein coupled receptors. We'll talk about them briefly, okay? But the important ones that are actually causing an electrical signal are going to be these ligand-gated channels. Yes?
Where do the vesicles and the neurotransmitters get packaged? Where do they come from?
Great question. So they get packaged up here, sort of further up in the head. And it's going to depend on which neurotransmitter it is, but there are going to be basically transporter proteins that are basically pumps that are attached to the vesicle that spend ATP to pack in the vesicle to concentrate that specific neurotransmitter into the vesicle. And every neurotransmitter has its own class of pump. Yeah. Now, an individual, so now here we go. So we've got our packed vesicles, right? We've fused here. The ligand or the neurotransmitter comes over here. It binds to these receptors. Okay. It's going to open an ion channel. Okay. In this case, this looks like it's a cation selective ion channel. And then we're going to have some membrane potential change in response to that. And then eventually, and it won't take that long usually, because these are usually relatively low affinity receptors, okay? The ligand will unbind, which will close the channel. We'll then return, postsynaptic membrane potential will start moving back towards its baseline. Okay. Any questions on this part? Yeah.
So you're saying that the calcium when it enters the cell, that's considered a substantial change in concentration.
Yep.
So there's so much that it is.
Exactly. Yes. Yes. Correct. Right. So with sodium and potassium, right? The thing is, you don't need that many ions to cross the membrane to really change that membrane potential quite a bit. Okay. So if you've at 150 millimolar outside and 15 millimolar inside, you only need to move thousands of ions across. Okay. And one molar is 10 to the Avogadro's number, 10 to the 23rd power, right? There's a lot of ions out there. Okay. Doesn't take much. But when you're talking about nanometer concentrations, or nanomolar concentrations, there, the number of ions that cross is actually significant to the concentration. Yeah.
I was going to ask, the different type of neurotransmitter affect the amount of this ion that is membrane permeability, and that's change intensity.
So does the type of neurotransmitter change the amount of membrane permeability?
What about the versatility within different types of neurotransmitters and receptors change the type of signals that it, and therefore what?
Right. So it will really come down to what does the receptor do, right? So the receptor could have different permeabilities to different ions, right? So you could have a receptor that passes only sodium ions or only potassium ions or only chloride ions. Not sure if I should get into it here, but I'll do it, okay? So the most common excitatory receptors are to glutamate. Okay, so glutamate's going to come in and bind onto here. Okay, now one weird thing here that confused me for many years, and if it confuses you, that's okay, is this glutamate receptor here, which is illustrated here. It is a cation. It allows positive ions to flow through it, potassium and sodium. But how does that cause an excitation? How does that cause a depolarization of the cell? Sodium's coming in, but potassium's going out. Why would that cause a depolarization? Shouldn't that be balanced? It's because of the impact that it will have on the membrane potential is really going to depend on where the membrane potential is before it opens up. Okay, so if you are sitting down at your resting membrane potential of negative, and this is kind of an advanced subject here, but you're sitting down at negative 70 millivolts, right? If you open up a channel that allows both sodium and potassium to pass through it, okay, how hard do you think the pressure, what we call the driving force on sodium to come in is versus the driving force for potassium to go out? Okay, potassium, okay, is pretty close to its equilibrium potential. Okay, equilibrium potential is like negative 90 millivolts. Okay, so if it's sitting at negative 70 millivolts, that electrical gradient is still pulling those potassium ions into the cell, and the chemical gradient is pushing it out, but it's not pushing it out that hard. Like the balance is the potassium wants to sort of stay around negative 90 millivolts, right? Sodium, on the other hand, okay, sodium is a positive ion with greater concentration outside. So the electrical and the chemical gradient are both pushing it in. So if you open up a ligand-gated ion channel for sodium and potassium, and you're sitting at negative 70 millivolts, then a lot more sodium is going to want to come in than potassium goes out. Okay, that's why you're going to get a depolarization. Now, as you get further and further closer to like 0 millivolts, all of a sudden that changes. Okay, that'll change it. In fact, at 0 millivolts, those two things will balance each other out, and you won't get any membrane potential change. Okay, so that's a little bit of a complicated thing, just as an aside. Okay, so let's talk a little bit more about calcium, because it's such an important signaling molecule, and it's important to many steps, not only in the release of the neurotransmitter, but also in the learning and memory and synaptic plasticity that we'll talk about in a little bit. How can we see calcium? Okay, how can we measure calcium levels? And if you'll permit me, I will go into a short diatribe on this because my thesis advisor got the Nobel Prize for inventing the technology. So here we go. Action potential. So this is the only place you're going to learn this stuff. Action potentials are going to cause calcium influx. Okay, we just saw that. An electrical action potential, right? That lasts for only a millisecond. Okay. But if there's voltage-gated calcium channels, they will open up and cause a flood of calcium to come into the cell. Okay. Now, the channels close very quickly. Okay. But the calcium's stuck inside the cell. So here's our membrane potential. This is the voltage change to an action potential. Here's a measurement of the calcium concentration inside the cell that was measured with a fluorescent dye that increases its fluorescence to calcium. Okay. So we see this increase in calcium. And then, so the increase is caused by the channels opening. Okay. When the channels close, the calcium level is stuck in there, and you have to turn pumps to pump out the calcium. So the calcium signal lasts for about a second or hundreds of milliseconds. So a single action potential can cause a doubling or tripling of the calcium concentration inside the cell for about a second. Okay. That is why it's an effective second messenger. Okay. So one thing that you can do with this, okay, is actually use it to look at synaptic activity and neural activity, because you can use calcium here, an elevation of calcium, as a proxy for when action potentials occurred. Okay. And this is going back to that. Remember that fruit fly that was green, the green fluorescent fruit fly? You can engineer that fluorescent protein by attaching it to some other molecules that we'll be talking about in the motor system, calmodulin, okay, and a peptide, and make it so when calcium's high, the fluorescent protein is green. And when it is not high, this is not bound, and the fluorescent protein becomes dark. You can then put this into a gene, or you can put this gene package into the brain using a virus, replace the skull with a piece of glass, permanent installation. It's called a window, glass crystal skull or window, brain window. Okay, this is done this is routine now. This was like very cool when we developed it like 15 years ago. Now it's just routine. It's still pretty cool. Okay, and then, okay, you can use a special microscope to watch the calcium level of individual neurons. So here, this donut
There's a neuron that should go there, and here's another neuron. We're looking at action potentials here, the red, and this is the fluorescence, which is in response to calcium. When action potentials occur. So every action potential causes a spike in calcium, which then drops back down slowly. You have a burst of action potentials, you get a huge increase in fluorescence.
So that's looking at calcium in a single cell, in the cytosol of a single cell. So action potentials in a cell, in the soma, will cause depolarization in the soma. They'll cause influx of calcium. Also, you'll see calcium at postsynaptic terminals coming in. Here's an example of each one of these little dots here is individual synapses getting activated. So these are the spines that stick out on the dendrites. And every time one of those lights up, that is a response to visual motion. This is recorded in the visual system of a mouse looking at different patterns moving. And the different directions of those patterns will turn on different synapses specifically. So individual synapses here have a particular preference for which direction a visual scene is moving in. And that preference is going to be inherited from the neuron, the presynaptic neuron that talks to it. Ultimately, we can use this to look at the activity of tens of thousands of neurons simultaneously across the brain. Each one of these blinks is an activity of a single neuron. And we're doing this via making them fluorescent and sensitive to calcium. And use calcium as a proxy for neural activity. Any questions on this? Okay, my aside is over. Now let's go back to the steps in this process. What do these different pieces look like? We've got synaptic vesicles, right? So this goes to your question, right? What do those synaptic vesicles look like? Inside, we've got 5,000 molecules of glutamate or GABA or something, right? Outside, we've got a bunch of different proteins that are directing it. This giant thing, there's usually about 2 of them on per vesicle. That's the transporter, okay? That is going to be packing in the neurotransmitter against its concentration gradient into this vesicle. There's a bunch of other stuff here. Some of that's involved in binding to actin filaments to like dock it. Other things are snare proteins that are going to latch on to other complementary snare proteins on the interior surface of that presynaptic terminal and allow the fusion to take place. They're going to pull themselves together and pull the plasma membrane into the synaptic. They're going to pull the synaptic vesicle into the plasma membrane and fuse it. Okay, so that's what our synaptic vesicles look like. What about on the postsynaptic side? How are we going to actually get an electrical signal on the postsynaptic side, right? We saw that we're going to open up channels, okay? And depending on the channel type, okay, what the channel is permeable to will have different impacts on the postsynaptic membrane potential. And these can be broadly classified into excitatory postsynaptic potentials and inhibitory postsynaptic potentials. We can, for short, we could call them EPSPs, excitatory postsynaptic potential, IPSP, inhibitory postsynaptic potential. An excitatory postsynaptic potential is going to move the membrane potential depolarizing closer to 0, closer to the threshold for firing an action potential. Remember, if we get over a particular threshold, maybe negative 50 millivolts or so, okay, voltage-gated sodium and potassium channels start opening up in the postsynaptic cell, we get an action potential. If we stay down below that threshold, we can move our membrane potential up and down, but no action potential gets generated, no signal will get propagated to the next cell in the chain until we get a sufficient size of this excitatory postsynaptic potential to go over the threshold for firing an action potential. So EPSPs, what causes the EPSPs? In the central nervous system, it's glutamate. It's the most common excitatory neurotransmitter, okay? In the peripheral nervous system, it's generally acetylcholine, cholinergic signals, okay? So onto the muscles, that's going to be acetylcholine. That neurotransmitter is going to open up cation channels that are permeable to sodium and potassium, as we saw, and that will allow depolarization to occur. Sometimes they're also permeable to calcium. With an IPSP, the most common neurotransmitter is GABA, which is a very small amino acid variant that looks like glycine, very small. And that is going to bind to chloride channels. Chloride reversal potential is pretty negative, around negative 80 millivolts. Okay, so you open it up, it's going to keep the membrane potential near negative 80 millivolts. Okay, so an excitatory postsynaptic potential will depolarize the cell. Inhibitory postsynaptic potential will hyperpolarize the cell. So, and we can modulate the permeability as well via G-protein coupled receptors that we'll see in a few slides. But for now, let's look at these potentials. Okay, now, an excitatory postsynaptic potential generally does not operate on its own. It's this local, we have a synapse, right? We saw our presynaptic terminal, okay? We have our postsynaptic side. The presynaptic terminal releases the neurotransmitter. We get a postsynaptic side. We open up channels right there, or right here, right at that active zone, that receiving end of that postsynaptic terminal. Okay. That's going to cause a local change in membrane potential, a local depolarization. That will spread, okay? As it spreads, it gets weaker, okay? But it will spread. So on all of those dendrites, right, you don't just have one synapse that's connecting onto a neuron. Remember, you know, on an average neuron that we saw sort of last week, we have like 10,000 synapses that are coming onto it, converging onto a single neuron. So you can imagine there's probably more than one synapse that's activated at any given time. Okay, if the neurons are always firing action potential, sometimes they're quiet. Most of the time a neuron's quiet. Maybe you use 10% of your brain. Well, that's not really true. I think it's more like maybe 10% of your neurons are firing at any given time. But you're going to use all of them. Okay. So you're going to have dozens to hundreds of synapses that are activated at any given second in your brain. Okay. And some of those synapses will be excitatory. They'll be glutamate-based. Some of them will be inhibitory. They'll be GABA-based. Okay? And the overall summation of their activity is going to be what controls the overall membrane potential of the cell and whether or not you trigger an action potential at this axon hillock. Okay, so these potentials add together in time and in space. So in this case, let's look at an activity of a single synapse here, this excitatory synapse. Action potential comes in, we get a release, we get an excitatory postsynaptic potential. It causes a depolarization of the cell. That depolarization doesn't get us all the way to the threshold to fire a neuron, and it only lasts for a few 10 or 20 milliseconds. Okay, then it decays back down as the channels close, okay, as the electrical signal fades away. So we can then fire another action potential, and we're going to get the exact same response. Okay. So these are sub-threshold fluctuations. They never get above to the threshold, okay? And they're spaced far enough apart that they can't really add up on top of one another. Yeah. Only the summation can cause an action potential. Yes. Yes. In almost any central nervous system cell, with very few exceptions, like the calyx of Held, will you be able to drive an action potential with a single synapse. Usually you need the concerted action of dozens of synapses working together to release simultaneously to trigger an action potential. So then this is sort of a toy example. This must be a very powerful synapse here, because so in this case, in this example here, we get two action potentials close in time. Okay, we get a depolarization, then maybe like 5 milliseconds later, we get a second release. So we get a second depolarization, and those two things can add up in time, cross the threshold and drive an action potential in the postsynaptic cell. This is called temporal summation, where if two releases happen within the period of time at which that EPSP lasts, they will add up on top of one another. Okay. Temporal summation. In this example, two of them in a row closely causes an action potential. In reality, it's going to take dozens. Okay. Also, we can add up in space. Let's say action potential one and there's two action potentials that hit two different synapses at the same time. Okay. Those two can add up together, okay, and cause a summation, a spatial summation, which can drive an action potential. And finally, we have the inhibitory postsynaptic potentials, these IPSPs that go negative, okay? They move us closer to negative 80 millivolts, the chloride reversal potential. Okay, so if we have an excitatory EPSP by itself, it looks like this. An IPSP by itself looks like this. Add those two things together. If you have an IPSP and an APSP happening at the same time, they kind of cancel each other out. So that summation of both excitation and inhibition is what's going to control the overall membrane potential of the cell. Okay. So you can imagine the only time an action potential in the postsynaptic cell ever generated is when there is a transient increase in excitation. More excitatory synapses are going off than inhibitory synapses. So if you get a lot of excitation and not much inhibition at a particular period of time, you'll tend to fire action potentials. If you get more inhibition than excitation, nothing's happening. So inhibition is going to subtract that excitation. Any questions on this summation? All right, so briefly, one slide, you can modulate the synaptic transmission. There are receptors on the postsynaptic side that are not ion channels. They're G protein coupled receptors. A neurotransmitter can bind onto that receptor, and instead of opening an ion channel directly, okay, it can activate a G protein, which then can cause the phosphorylation or the change in sensitivity of other ion channels and other receptors on the postsynaptic membrane. Okay, so that can make it easier to cause a depolarization. It can do a variety of different things to sort of tune the overall strength, the overall sensitivity of that synapse. And those changes don't last they take seconds to implement because there's a whole chain of events that have to occur for that to happen, and they last for seconds to minutes. So this is modulated synaptic transmission or metabotropic receptors, G protein coupled receptors. So what are our neurotransmitters? There's a whole variety of them. Acetylcholine. They're all pretty much small molecules. Okay, we've got acetylcholine that is going to be excitatory on skeletal muscles, excitatory in your central nervous system, inhibitory on the heart. Okay. We've got norepinephrine, dopamine, and serotonin. These are all pretty structurally similar, right? Norepinephrine and dopamine just differ by one particular hydroxyl group. Okay. And these can have a variety of effects, excitatory or inhibitory, on the central nervous system. They're thought to be neuromodulators in general. So they can sort of tune up or tune down synapses, okay, by their release. They can also trigger excitation on their own, but these are really going to modulate particular pathway brain, overall general sort of activity of the brain. Then we have our amino acid receptors or neurotransmitters. The two big ones here are GABA. This is the primary inhibitory neurotransmitter in the brain, and glutamate, the primary excitatory neurotransmitter in the brain. Glutamate's also, there's receptors for glutamate on the tongue, monosodium glutamate. That's great, makes things taste better. Right? That is just a receptor for glutamate that's causing a depolarization on the tongue. We have glycine. It's inhibitory. We don't talk about it that much. Then there's a bunch of neuropeptides, many of which are not even discovered yet, which are the sequences of short chains of amino acids that will bind to particular G protein coupled receptors that are going to act to, usually they're linked to some specific drive, right? They influence pain, or they influence like certain motivational states, thirst, hunger, things like that. Molecules that mimic them, like GLP-1 agonists, right? Ozempic, can have profound impacts on motivation and behavior. And then finally, we have this gases. Nitric oxide is considered to be a neurotransmitter, okay? And not only can it do things to vasodilate, cause vasodilation, but it also can change the probability of releasing vesicles when an action potential comes in. Okay, so let's look in a little more detail at glutamate. So this sort of gets to your question, how do you sort of fill those vesicles up? Okay, so here is a glutamate. Our glutamate molecules, they get released. They can come across here and they can bind onto a variety of different channels. You don't need to know all these channels, just to show you that there's a variety. There's these NMDA receptors, AMPA receptors, kainate receptors. These all bind glutamate. They're not named. These are all glutamate receptors. Okay. They're just named different things because when they're trying to figure out, oh, what is actually on here, they would put all these different drugs on, okay? And when they found a drug that specifically activated one channel, but not any of the other channels, that's the name of the receptor that they called it. They called it after a drug that will specifically activate just that channel, even though they all are activated by glutamate, okay? So we've got these NMDA and AMPA receptors are the sort of most important ones. Metabotropoglutamate receptors binding to this is going to modulate the activity of those through G proteins. Okay, so then we're going to have these astrocytes that sort of scavenge up glutamate that leaks out through these transporters, then sort of change it into other forms, pass it back into the neuron, and the neuron will repackage it into synaptic vesicles. Now, drugs, right? Any psychoactive substance, whether it's an anesthetic or a stimulant or anything like that, is always going to be acting on receptors and mimicking neurotransmitters or modulating the activity of receptors that bind to neurotransmitters. So the action of any drug is going to be through that. Okay, so one of the sort of, I guess, a drug that we use for anesthesia in animals, ketamine, that is going to bind to the NMDA receptor and block it. Okay, a lot of other sort of drugs that influence the NMDA receptor cause dissociation. Yeah. Then, so those are sort of antagonists that block those receptors. Glutamate receptor agonists generally are never sort of used because they will just cause seizures. Right? If you're activating there's a lot of glutamate receptors in the brain, you're just going to cause a seizure if you apply any of those. Then we've got GABA, or this is the GABA receptor here. GABA binds to it. It causes chloride to come in, but alcohol modulates GABA receptors. It's going to change your behavior, and depending on the level of alcohol, is supposed to be an inhibitor, right, or something, but really it's sort of activating these inhibitory receptors. But where the receptor is in the brain and what circuits it turns on or turns off is really what is going to influence the behavior and influence the psychological response to that. You can mimic GABA with other compounds like GHB, which then can just knock people out. Many actors have died from using GHB in concert with alcohol. Okay, acetylcholine. Acetylcholine is a very common neurotransmitter, right? It's going to do a variety of things. We talked about it's going to activate our muscles, right? It also, in the central nervous system, controls where attention goes. Where do you turn your spotlight of attention? That is going to be determined by the overall pattern of acetylcholine release in your brain, okay? So acetylcholine has a variety of different receptors. This one is called the nicotinic acetylcholine receptor. Can we guess why it's called a nicotinic receptor? It's because nicotine is a substance that will specifically activate this acetylcholine receptor and not any other, and not the muscarinic acetylcholine receptors. So this is why cigarettes have a function, right? They have some sort of focusing function, or Zins or whatever, right? A focusing function that is dependent on the action on nicotinic acetylcholine receptors. That action is also why it reinforces in an addictive manner. Then we have serotonin, dopamine. These are drugs. So dopamine is sort of a molecule that both of these molecules are produced in very small sets of neurons that project axons all across the brain. Okay, so in the case of dopamine, dopamine is going to be the signaling molecule and the neurotransmitter that's most tightly linked to reward. And it will signal to specific brain circuits to strengthen in response to its presence. Okay, so the release of dopamine is a reward signal. When something good happens to you, okay, or when you anticipate something good, and eventually when you anticipate something good happening to you, dopamine, the release of dopamine is the signal that shows that matches that expresses that reward, and is the signal to the brain to change the circuitry of the brain to try to do whatever just happened in that circuit. Okay, so dopamine on its own is going to reinforce, internal dopamine is going to reinforce, actions that you want to take place, that rewarded actions in which we received a reward. You're going to want to keep doing those because of the release of dopamine. Serotonin, on the other hand, is really more of it's sort of a molecule that's more involved in relaxation, and influences sleep and mood, memory. And serotonin projects all over the brain, and it increases quite a bit during sleep. It also is involved in sort of emotional attachment. So one of the cool things here, one brief aside again, got to do it because it's pretty cool. So acetylcholine, actually, when I said that you can modulate the spotlight of attention, right? You can only really think about, it's really hard to attend to more than one thing at a time, right? Sometimes you can do it, like if you're driving, right? And you can go on autopilot, you can listen to a podcast at the same time. But if you're trying to write a paper and listen to a podcast, forget it, it's impossible, right? So this is another thing, this is engineered in our lab in collaboration with another lab. You can now finally, for the first time, be able to watch where acetylcholine is released in the brain, okay, in real time. This is a cool experiment. We put that window on the mouse, okay? We made a fluorescent protein that changes its fluorescence in response to the presence of acetylcholine. And now we're going to watch as the mouse is sort of thinking and doing stuff, moving its whiskers and stuff, and see where the acetylcholine is released in the brain. So these frontal areas are areas involved in motor planning. So when the animal's moving its whiskers, it's starting to tend to plan. I'm going to move my whiskers. I'm going to try to investigate something. We get a release of acetylcholine up here in the frontal part of the brain. When it's listening to other sensory information, it's going to get acetylcholine released further back in the brain. Anyway, this research is still ongoing. Got a couple undergrads working on it. Okay. Serotonin. Let's talk a little bit more about serotonin, because serotonin is really important for depression, right? Many of the most effective drugs for depression, Zoloft, Prozac, things like that, those drugs operate through modulation of serotonin pathways and modulation of serotonin-based receptors. So many of the antipsychotics, any sort of chemical that is involved in modulating mood and modulating behavior, many of those things are going to be activated through serotonergic pathways, so serotonin receptors. And you don't have to memorize something like this. But what I'm showing you here is that different neurons in the brain, these are all different this is slices of different parts of the brain, different areas of the cortex of the brain. Okay. And what we can see here is different types of serotonin receptors, okay? So there's not just one serotonin receptor out there. There's many. There's a whole family of them, okay? And they are expressed, here the black spots tell you what neurons they're expressed in. So this A lot of 5-HT1A receptors over here all over the place. These 5-HT2As are down here in the middle. These 5-HT2Cs are kind of rare, sort of sprinkled about, right? All of these dark neurons are expressing different patterns of that serotonin receptor. Okay. So a drug that mimics the structure of serotonin does not have the exact shape of serotonin. It might have a shape with a little thing sticking off of it, okay? That will allow it to selectively activate or suppress different ones of these receptors. All the receptors bind serotonin, but you can design drugs that will specifically activate or inhibit only certain classes of those, which then allow a targeted modulation of specific neural circuits. Okay? So in particular, you have, in particular, the action of Prozac and other serotonin selective reuptake inhibitors is accomplished through changing the transporter here and influencing the transporter to be less effective and allow serotonin to persist in the synaptic cleft for longer periods of time. That then causes some sort of adaptive change in the expression of other receptors here that will drive a change in mood over the course of an immediate change in mood, and then a more prolonged change in mood after a few weeks after the brain sort of tries to maintain homeostasis. So what about dopamine? There's many stimulants, right? All stimulants are going to mimic the structure of dopamine or the structure of norepinephrine because they're very similar, okay? So things like Adderall or amphetamines, those are all going to mimic dopamine and either cause the blocking of the reuptake of dopamine, so increased persistence of dopamine in the synaptic cleft. It gives you more sort of reinforcing and more sort of energy to focus on a task, or the even reversal of the dopamine transporter to dump dopamine out, even in the absence of synaptic transmission. So you can imagine that might be very powerful if to reinforce specific behaviors also can drive addiction, right? Because of inappropriate release of dopamine to actions that are, driving dopamine release can lead to behavior that seeks out those drugs that will enhance dopamine release. Yeah. Is that an autocrine signal? Good question. No, I wouldn't call it an autocrine signal. Because it's so the drug is acting here on the presynaptic side, but the actual action of the dopamine on the neural activity is going to be on the postsynaptic side. Wouldn't call it autocrine. Then we have things like neuropeptides and opiates, right? So a lot of the pain signaling in your body, all the pain signaling is really accomplished through these peptides that are released, substance P, a bunch of these different peptides. And endorphins are naturally occurring molecules that will interact with these opioid receptors, block them in order to provide pain relief. Okay? So many of the most powerful opiates or opioids that are very powerful in relieving pain are doing it through binding onto those opioid receptors and mimicking the actions of the endogenous endorphins. Okay? Now, that, of course, I think in the last decade, we've seen a huge increase in the amount of drug overdoses due to opioid abuse. And it really comes down to, I think, development of pharmaceuticals like OxyContin, which were very powerful time-release things that provided potent binding to opioid receptors, got many people addicted to it. And then the production of fentanyl, which was a much more potent version of these opioids like morphine or heroin, right? And so that the sort of here we have like an example of a dose of heroin that would kill a person. There's also fentanyl that would kill a person. So you can imagine, right, if there's a drug dealer that was taking fentanyl and trying to mix up with a bunch of, I don't know, baking soda or something, right? They don't mix it properly, okay? You get too much fentanyl in one bag than another bag, you die. Oops. And fentanyl is also it's so potent that it's very difficult to overcome that binding. So it's harder to displace from the opioid receptors than a normal, opioid. So things like Narcan have a more difficult time to cure that. So thankfully, they've developed immunoglobulin technology has developed test strips to identify that in recreational drugs, which is great. Hopefully that will reduce deaths. Okay. So how do drugs drive addiction? Okay. It really is, and this is, a very simplified sort of slide about how it does it. Okay. But it's going to cause some change in neuroplasticity, right? You're going to have to strengthen some pathway that wants you to drive your behavior to take that same action again. Okay. So whatever, and there's different drugs will do that in different ways. Okay. So if you have some inhibitory drug, like an opioid, opium, heroin, fentanyl, that can act and inhibit a neuron, which inhibits this other neuron. This neuron is a neuron in the ventral tegmental area. It's a part of the neural circuitry that sort of controls your motivation to do specific things, provides the rewards and the intrinsic motivation to do particular things. Okay, so activity in the VTA, when it's associated with an action that you're taking, will tend to drive you to take that action again. So if we increase VTA activity, we're going to tend to do that action again. Nicotine directly will increase VTA activity. Opioids will inhibit a neuron that inhibits VTA activity. So indirectly enhances VTA activity. And then VTA activity over here, it's projecting onto these other neurons in the reward pathway, and stimulants, okay, are actually going to upregulate the release of the VTA activity onto these reward system neurons. Okay, so 3 different areas, right? Either blocking inhibition of VTA neurons, increasing the activity of VTA neurons, or increasing the release of presynaptic terminals of VTA neurons. Now, all of those things, right, that activity has to then, in order to accomplish a change in behavior, you have to accomplish a change in the physical structure of the brain. We have to make some synapses stronger and some weaker, right? There's a process called neuroplasticity, okay? And there's sort of 2 ways that you can do neuroplasticity, okay? So during development, we're born with double the neurons than what we're going to have after 2 years. Those neurons are going to try to wire up together and compete for resources that are released by electrical signals that are different neurons talking to it. The neurons that are connected, that most effectively communicate between one another through synapses, will tend to strengthen those synapses and strengthen those neurons. Okay, they're going to compete for these growth factors and activity. The cells with the highest activity are going to be wired together. The cells that don't get enough activity are going to kill themselves, commit apoptosis, and give their resources to their neighbors. This is a part of activity-dependent development. Okay? And that allows us to wire up more intricate patterns of neural circuits than you might otherwise have if you had to specify everything through genetics. Now, the term neuroplasticity is after birth, okay? How can we strengthen or weaken specific synapses or specific neural pathways, okay? So, how do we delete this synapse and add this other synapse here, or make this synapse stronger or this synapse weaker? It is all going to be dependent on the coordinated activity of the presynaptic terminal and the postsynaptic terminal. In the simplest form, which is a rule called Hebb's rule from Donald Hebb, who formulated it in the 50s, is that cells that fire together, wire together. So cells that are coactive and are synaptically connected will tend to strengthen those connections. Cells that have one side active while the other side is quiet, where one side cannot sufficiently drive the postsynaptic side, those connections will tend to get weakened. Okay. So how physically can a cell determine whether or not the presynaptic side and the postsynaptic side are both firing action potentials together? You have to have a presynaptic side that fires an action potential, releases neurotransmitter, and then shortly afterwards, the postsynaptic side has to fire an action potential. If that takes place, that synapse will get strengthened. How might that happen? So the process of long-term potentiation. Okay, it's going to involve those glutamate receptors that we talked about, that we mentioned on that slide, AMPA and NMDA receptors, and it's going to involve calcium. Except this time, the calcium is not just in the presynaptic side, it's also being used as a signal for strengthening a synapse in the postsynaptic side. So you can imagine our presynaptic neuron here. Prior to long-term potentiation, it has some receptors on it, okay? And the receptors come in two classes, okay? You have AMPA receptors and you have NMDA receptors. AMPA receptors pass sodium and potassium. Okay. If you activate them, you'll get a depolarization. And NMDA receptor passes sodium, potassium, and calcium. So if you activate an NMDA receptor, you'll also get a depolarization. But in addition, you will get calcium that comes into the cell. So if NMDA receptors get activated, calcium comes into the cell, and that calcium will serve as a signal to strengthen that synapse. So sort of the process here is if we insert AMPA, so if we activate these NMDA receptors, okay, and calcium comes into the cell, that calcium will mobilize AMPA receptors to insert themselves in the membrane, create more glutamate receptors in the postsynaptic side, and then make the postsynaptic side more responsive to the same amount of glutamate that's released. If you've got more receptors on the postsynaptic side for a thing, then you're going to get greater depolarization, right? When that glutamate's released, because we're going to have, you've got more than enough glutamate that's released, okay? 5,000 molecules, you've got hundreds or dozens of receptors on the postsynaptic side. Double the number of receptors, you're going to pretty much double the strength of your postsynaptic depolarization for a single vesicle release. So why isn't everything getting stronger all the time? Okay, if we activate these NMDA receptors, like why we should be getting this potentiation all the time? Well, there's one specific aspect of a NMDA receptor that also differs from the AMPA receptor. Okay, and so it passes calcium. That's one thing, but it also has a block in it. It's normally blocked by this molecule, magnesium. We haven't talked much about magnesium. We've mentioned it like it's out there, right? We haven't really mentioned what magnesium does. Magnesium's job is to block the NMDA receptor whenever the membrane potential is very low, whenever it's hyperpolarized at -70 millivolts. Okay. If this side of the cell, okay, this postsynaptic side has at -70 millivolts, it's down here at a resting membrane potential, and glutamate comes in and binds to the NMDA receptor. Guess what? This magnesium molecule is positively charged. It sticks in the pore of the NMDA receptor, and even if a glutamate binds onto it, the receptor will open up, but it will be plugged by this pore. So no ions can pass through an NMDA receptor unless the magnesium is removed. How can we remove the magnesium? If we were in a dish, we could just like wash it away, but that's not, we're in a brain, we can't do that. So instead, we have to remove the magnesium from the pore. What's pulling the magnesium into the pore? The negative membrane potential. It can sense that negative potential, okay? So if you reduce that amount of negative potential, the magnesium will fall off the NMDA receptor. So the NMDA receptor, if you have a depolarization on the postsynaptic side and the presence of glutamate, okay, that's when NMDA receptors can actually open and pass current. So it has to be a combination of the postsynaptic cell already has to be somewhat depolarized to allow the NMDA receptors to finally open up in the presence of glutamate. And that is what's going to cause the conjunction, this ability for the presynaptic neuron to know that the postsynaptic neuron is activated. They can send the signal. They'll send calcium in only when the presynaptic and the postsynaptic neuron are co-activated. The presynaptic neuron has released glutamate. The postsynaptic neuron is depolarized from maybe other synapses nearby or from an action potential that just happened. And that will allow the magnesium to temporarily come off. If glutamate's around, then ions will flow in, not just sodium and potassium flowing, but also calcium. That calcium is what is going to drive the potentiation of that synapse. Specifically, there's a couple ways we can do it. Calcium is going to hit these. So calcium comes in through the NMDA receptors. Calcium does not come in through AMPA receptors. Normally, there's a lot of AMPA receptors at these synapses. Remember, AMPA and NMDA receptors, they both bind glutamate. They're only labeled because AMPA is a drug that activates AMPA receptors. NMDA is a drug that specifically activates NMDA receptors. So you bind on, glutamate's released, it binds on. If there's no magnesium block, okay, we had maybe some depolarization here, okay, then calcium can come in. It's going to activate some signal transduction pathways, bunch of second messengers, and it can strengthen the synapse in 3 ways. Way number 1, it can phosphorylate an AMPA receptor at a specific location that causes the AMPA receptor to increase its conductivity. It allows more ions to pass when it opens up. Okay. Number 2, it can interact with vesicles that contain AMPA receptors and cause those vesicles to migrate and fuse with the postsynaptic side and increase the number of receptors on the postsynaptic side, making that postsynaptic side more sensitive to synaptic release. And 3, it can activate the synthesis of nitric oxide. Nitric oxide is a gas. It is easily diffuses through plasma membranes. And there are receptors for nitric oxide in the presynaptic terminal that will increase the probability of synaptic release when an action potential comes in. It'll increase the number of vesicles that get released. So you could have a postsynaptic changes by making existing AMPA receptors more sensitive. You could have the addition of additional AMPA receptors to the membrane. And you can even have this sort of backward signal to the presynaptic side that is going to enhance the amount of glutamate that gets released. Okay. So that's long-term potentiation. If you have presynaptic and postsynaptic activity simultaneous with one another, you'll get sufficient calcium in to drive the potentiation of that specific synapse, right? Only the synapse is going to get strengthened. If you have to have the synapse, both sides have to be activated. We can also cause a long-term depression. We have to have a way to weaken the synapses over time. Otherwise, we're screwed. Every synapse is going to get stronger eventually. We have super synapses everywhere. That is not good. We want to have a balance of activity in the brain. You always want to maintain homeostasis so that the total amount of activity, the total amount of synaptic strength is equal across the brain. Just some are getting stronger, some are getting weaker. That's how we're going to form memories. So for long-term depression is, so here's a little plot that shows our long-term potentiation, right? We've got calcium coming in through the NMDA receptors. It turns out if you activate a presynaptic terminal repeatedly over and over and over again, and the postsynaptic side never fires, occasionally you get a little bit of, occasionally the magnesium won't be fully attached to the NMDA receptor. You'll get a little dribble of calcium inside the postsynaptic cell. If you just get a little bit of calcium that comes in the postsynaptic cell, that actually triggers the internalization of AMPA receptors and the shrinking of the postsynaptic terminal. So that's called long-term depression. For that to happen, basically you have to have repeated activity in the presynaptic terminal that is never aligned with activity in the postsynaptic side. All right. I know that was a ton of information. Thanks for paying attention. And I'll do a Q&A at 11:45 on Zoom, any topic you want, and that'll go for about an hour. All right. See you Thursday.
One more to go.
Yeah, buddy, I'm ready.
So Thursday, we're gonna have the second exam.
Okay,
So if
okay.